Okay, so welcome back to this third video on the phosphoinositide pathway. So I'm aware that I just gave you a basic description of the phospholipid bilayer composition in the previous video. Now what we're going to do is actually see what PIP2 is. Right, okay, so PIP2 or phosphatidyl inositol 4,5-bisphosphate is a molecule which you find in the phospholipid bilayer, and I want to show you how it's just basically a modified version of a phospholipid, basically. Okay, so before we discuss what its actual structure is, I firstly just want to tell you what the molecule inositol is, because as you can see, phosphatidyl, you should hopefully have some idea what that is. I've just told you what the molecule phosphatidate is. So basically phosphatidyl just means that you have a phosphatidate molecule in there somewhere. So now what we need to turn our attention to is what is inositol. So inositol then. Inositol is a 6-carbon ring, basically. So it's a hexagonal ring of carbon, like so. And basically, off each carbon, you have a hydroxyl group. So you have 6 hydroxyl groups in total, 1 off each carbon, like so. Here, here, another hydroxyl group off this carbon, and another hydroxyl group off this carbon. And then each carbon also has a hydrogen coming off it. So a hydrogen off here, hydrogen off here, a hydrogen off here, hydrogen off here, and a hydrogen off here. So that is the structure of inositol. That's pure inositol. Now we're in a position to build phosphatidyl inositol 4,5-bisphosphate then. Right, so let's start with phosphatidyl then. Actually I want to keep this name in sight. So we're going to start off with this phosphatidyl clue. So that tells us that we need a phosphatidate group in here. So let's draw glycerol, and I'm going to draw it on its side this time so that I can fit it all in and keep the name in sight. Okay, so to get phosphatidate, what you have to have is you have to have a phosphate group linked off the third hydroxyl group of your glycerol molecule. Then you have hydrogen here, hydrogen here, hydrogen here, and then of this hydroxyl group we know it's esterified to some long-chain carboxylic acid. And I want to stress that it's not necessary to know what these long-chain carboxylic acids are. A phosphatidate molecule, it's not just one molecule, it's a family of molecules where you just have long-chain carboxylic acids in these positions. So it's not a specific long-chain carboxylic acid that you have to have in these positions. It turns out that usually there is a specific long-chain carboxylic acid. Certainly in this second position it's usually arachidonic acid, but that's a different story. Okay, so here we have phosphatidate. So phosphatidyl, basically what you do is you take off this hydrogen and you link that to some other group, and this whole thing here is then the phosphatidyl group. So this entire structure, this phosphatidate molecule, now that you've just taken that hydrogen off and you're linking it onto something else, this is the phosphatidyl group, and I shouldn't be labeling it over there because I'm going to draw the rest of the molecule over there. So this is this phosphatidyl group that we are adding on to something. It was the phosphatidate molecule, now it's the phosphatidyl group. And what are we linking it onto? Well, we're linking it onto inositol. So we're linking it onto inositol. And basically what we're doing is forming another ester link, basically. So we're taking the hydroxyl group off of this phosphate group. And we're taking the hydrogen off here, and we're bonding that oxygen to the phosphorus. Okay, so now we've got phosphatidyl bonded to our inositol molecule here. So here's our inositol. And each of these has a hydroxyl group off it. But we also need to remember that this isn't just phosphatidyl inositol we want to create. This at the moment, what we've created, by the way, at the moment is phosphatidyl inositol. We've linked the phosphatidyl group to an inositol molecule. But we want to create phosphatidyl inositol 4,5-bisphosphate. However, let me just highlight the bits of the name and label everything up before we go any further. So this is the phosphatidyl group there, that in pink. Now, this group here that I'm adding on here is this inositol molecule that I've just described to you. So that's inositol here. And now we want to just add on 4,5-bisphosphate. So we just add on phosphate groups to the 4th and the 5th hydroxyl groups of this inositol molecule. Now, this is the confusing thing. Why isn't this 3,4-bisphosphate? Because if you look at these carbons, 1, 2, 3, 4, 5, 6, but it's completely symmetric. What's the difference? Why would you call one of these 3 and one of these 5? Well, you have to choose one to be 3 and one to be 5, but the reality is that there isn't any difference between them. So what you're basically going to do is you're going to add a phosphate group onto this one. That's obvious, but you're also going to add a phosphate group onto this one. But why wouldn't you call that phosphatidylinositol 3,4-bisphosphate? I don't know the answer to that. But what they do basically is they take that hydrogen off and they do indeed put phosphate groups on here, like so. So here are the phosphate groups on the 4th and the 5th carbon. Okay. But as I say, I don't understand why you don't call that phosphatidylinositol 3,4-bisphosphate. It would make more sense to me, but it's perfectly legitimate to call it phosphatidylinositol 4,5-bisphosphate. So these phosphate groups are that final, portion of the name basically here. So these make up this 4,5-bisphosphate. Right, okay, so now this entire molecule is phosphatidylinositol 4,5-bisphosphate. Right, and basically, as you can see, it is just basically a phospholipid with a few extra groups stuck on its polar head, basically. And what phospholipase C is going to do is it's going to take this molecule, which is in the phospholipid bilayer, so you do have this sitting alongside the normal phospholipids. You do have this molecule in your phospholipid bilayers, and basically what phospholipase C is going to do is it's going to cleave this bond here. It's going to break this bond up here, yes, it's going to break this bond here, and what that does is it breaks off this great chunk of a molecule, this inositol with 3 phosphate groups, and then it also gives you a glycerol bonded to 2 long-chain carboxylic acids. So, we know what a glycerol molecule with 2 long-chain carboxylic acids is called. That's called a diacylglyceride. So this gives a diacylglyceride. And so all we need to do is name this molecule of inositol bonded to 2 phosphate groups, which is the product of this phospholipase C beta enzyme. And basically, that's inositol, so we're going to name it inositol something. And then we've got 3 phosphate groups, so we'll just call it inositol 1,4,5-trisphosphate. Inositol 1,4,5 trisphosphate. And that's a bit of a mouthful, so we'll often just denote it I for inositol, and then P for phosphate, and then 3 to denote that we've got 3 phosphate groups, so IP3. Okay, so these are the 2 products of the enzyme phospholipase C beta, which was activated in response to our G protein coupled receptor being activated by its ligand. Right, now, IP3 basically goes and activates a receptor that we are going to study in a lot of detail in our next coming up videos. It basically activates an enzyme which is placed in the endoplasmic reticular membrane. So if this is the whole cell here, then if we draw the endoplasmic reticulum as an organelle within this cell, then basically there is a receptor in the membrane of the endoplasmic reticulum known as the IP3 receptor. And what this IP3 receptor does is when IP3 binds to it, it doesn't open. That's a misunderstanding. It doesn't open in response to IP3. What happens is that it gets it changes conformation, and it gets into a state where it can be opened. But the ligand that actually opens it isn't IP3 at all, it's calcium. Calcium actually opens the IP3 receptor. What IP3 does is that it changes the conformation of this receptor so that calcium can actually bind and open the receptor. So until IP3 comes along and binds there, the receptor cannot be opened by calcium, and in fact it's actually it's inhibited by calcium. What IP3 does is it changes the conformation, and it makes available a ligand binding site for calcium that's actually going to activate the IP3 receptor when calcium binds, and make the IP3 receptor more likely to adopt an open conformation. And when the IP3 receptor adopts an open conformation, it will allow calcium to move through it, and calcium will leave the endoplasmic reticulum and go into the cytoplasm, basically. So this is the IP3 receptor here, and we'll study that in more detail later. Right, for now, what I want to discuss is how IP3 is actually degraded, basically, and how it's returned, how you remake PIP2, basically, because so far we've seen how the phospholipase C-beta breaks down PIP2, but we haven't seen how you actually make it back again. So basically what happens is that if we use this, in fact actually we'll get a new piece of paper and we'll draw IP3 out again. So we'll do that in the next video.